And we're off. So hopefully I'm safely in San Diego as you're listening to this. I just dropped my phone, which has the timer on it. And hopefully you've remembered that this is a flipped week where the quizzes are occurring on Thursday, both the read ahead and the weekly. And today will be the major lecture. And I find it very odd to lecture to myself and a computer, but what can I do? So if we start at the course homepage, love that picture, and take a peek at the calendar, it should show this is election day, sort of, so it's emotions and stress. And as it says, Thursday read ahead, and actually, I want to lecturing today and last week. So other than that, everything is pretty normal. I have created a page for this, which is linked over here somewhere, November 6th lecture, which I already have open over here, which includes some resources that, unless I say otherwise, I'm not requiring, but I'll be referring to this page as I do the lecture and supplementing it. And this, by the way, this chapter is relevant to my research. We're going to revisit opioid and non-opioid at some point this week, if not today, on Thursday. And it's pretty related to the faculty talk that Clavius, this faculty discussion I'm dealing with by Sapolsky, about Sapolsky's book, Behave. And it's also relevant to his earlier book, Why Zebras Don't Get Ulcers. He's very big on the effects of short-term and long-term stress on the body. Short-term is not so bad. Long-term, in general, is pretty bad. Let's take a look at the textbook. So the opening vignette, and then later in the chapter, he returns to this. This is a woman who feels no fear, and given what happened in this opening vignette, she's kind of lucky to be alive. Interesting that her interpersonal space seems narrower than most, and that must mean I'm very fearful because I have a giant interpersonal space zone around me. The amygdala figures prominently in this chapter's subparts of the amygdala, inputs and outputs of the amygdala, including arguably my favorite part of the brain, where I did the electrical stimulation produced analgesia, opioid and non-opioid, which I'm obsessed with, the periaqueductal gray, or PAG, in the midbrain around the cerebral aqueduct. That's in here. So it's a good chapter, and I feel bad that I'm not there with you when we're doing this. There's a discussion of what are emotions and how many kinds of emotions there are, and some classic theories here that you have to know. James Lang is one of them. William James is a hugely important figure in American psychology, certainly psychology in general. This table contains no new information, as they often don't. On top, you have folk psychology, common sense, the kind of common sense that tells us the earth is flat. And you should know that sequence. Then there's the James Lang theory, and you should know that sequence. And then, canon, the famous canon, the dead canon, Cannon-Bard theory, which has a somewhat different perception on things, and notice that after the perceptual integration danger component, the pathways split into autonomic responses and specific emotions. So be able to compare and contrast those, recognize the patterns for the three different kinds of theories, and we'll see that there's one more to come. Schachter, usually Schachter and Singer, cognitive attribution model is sort of like Cannon. And there's a picture on the next page. Notice epinephrine is mentioned here. And we haven't talked very much about epinephrine. It's a synonym for adrenaline. And of course, it's related to norepinephrine, which is a catecholamine. You might take a look at the synthetic pathways, which are part of Rolling Thunder anyway, because they may come up again here. And there's a classic study that the book doesn't seem, and there we go, Schachter Singer is down here. A classic study prior to the one that's discussed here, where people, male people, cross a suspension bridge very high above a gorge or close to the bottom of the gorge. And if you go across the high suspension bridge, presumably you're experiencing autonomic arousal from the kind of scary experience, whereas when you go across the lower bridge, you're not so autonomically aroused. At the end of the bridge, a female comes up to you with a clipboard and says, "Would you like to participate in some research that we're undertaking?" And you presumably say yes. Questions are asked. Answers are given, and then the investigator tells you, because the study is currently ongoing, I cannot tell you right now what the study was about, but here's my contact information. And after this date, I can tell you about the study. So if you want to know about the study, contact me using this information after this date. And of course, that's what the study's about. So you're a male, you run into a female after going across a high bridge or a low bridge, and she gives you her contact information, and the manipulation, which is not a manipulation because you chose which bridge to go over, was the bridge and the autonomic arousal. And the question is, which kind of male is more likely to contact the investigator after going over the bridge, the one that went over the high bridge or the low bridge? And the statistically significant tendency was that the men who went over the high bridge were more likely to call her back, suggesting, as you'll see in the Schachter-Singer model, that the autonomic arousal they were experiencing when they ran into the woman fed into perhaps a perception that they were attracted to her and they would like to talk to her again. And in ancient Rome, they were giving dating tips to ancient Romans who weren't ancient, but they were normal aged and they were dating. And it was suggested that if you wanted your date to go with you, you should take them to the Colosseum, particularly if there was slaughter going on that day. The woman would sit there with you watching slaughter, getting autonomic arousal, and they turn to you and think, "Oh man, this guy, I really like him." Of course, the modern version of this, particularly around Halloween, is to take your date to a scary movie, which induces autonomic arousal. And if you live in LA, you might go to Thunder Mountain or something and get on a roller coaster. And you're hoping that the autonomic arousal brought to you by the roller coaster or the haunted house or the movie will be misattributed to you, and you look to her to be more attractive. Now, in this study, which is an experiment, because they're actually manipulating both the drug you're receiving and the confederate you're put with. So you get epinephrine, adrenaline, or not, and you are put with a happy confederate or an angry confederate, and the hormonal arousal is the same, but one person is modeling happiness and one person is modeling anger, and oh wow, you end up being unusually happy or unusually angry. So there's a figure that really doesn't say anything new, but it should help you um, understand the Schachter-Singer model. Polygraphs, lie detectors, hope that um, your freedom is never dependent on this one. My brother used to be in a federal agency where they would give these things randomly. He was always, anxious that some operator would decide he was lying when he wasn't. And here's images from Darwin, 1872, expression of emotion in humans and animals. Um, he was interested in faces, not the way I'm interested in faces. I want to look at the bone structure. But Darwin was interested in the evolution of emotional expression and whether, facial stimuli or facial signaling occurred in species other than us. And there's tickle faces, and you can tickle rats, and it's fun. Um, and then there's this is an interesting study about putting alcohol on infants and seeing if they're very reactive. And reactive infants do seem to grow up to be reactive adults, and probably should receive some sort of special attention to try to keep that from happening. I'm not going to test you on this figure, but I'd like you to take a look at it. Ekman over here, early on in his research, I was pretty impressed with Ekman. He he learned all the muscles in the face, and there are a bunch of them. And when they contracted and tried to look at emotional expression associated with muscular, therefore neural activity, and he did good science. Lately, he's gotten into micro expressions and recognizing lying, and is trying to sell products to Homeland Security and the FBI and other people to recognize when lying has occurring during interrogations. I'm less impressed with his more recent stuff. It's turned into a commercial enterprise for him. He's not really publishing a lot of science these days. And here are some classic faces, and for some reason, this happiness, every time I see it, I feel like I know that person, but I guess I don't. And that's worth looking at in terms of this would interest Darwin. Are emotional expressions of different people be recognized by different people? And the answer is sometimes yes, and the answer is sometimes no. So take a look at that, and certainly know the outliers where emotions are difficult to express or to recognize in isolate populations. This figure has occurred in like 3 editions. Interestingly, in the previous 2 figures, the human there was the president of the United States. And in this edition, it's not, although she did win the popular election. I have to wonder whether this book went to press prior to knowing who won the election. But anyway, so that's a figure they always use in here. There's your face peeled back and nerves innervating it, and you can see the facial nerve, superficial, and superficial gives you a lot of micro expressions and stuff. And you can see where if this nerve were damaged, you would have some trouble with emotional expression. This research, this may not be replicating. The research was that if you give a human a pencil and tell them to hold it in their mouth, and they bite it, it makes them make a smiley face, and then you evaluate their emotions and they are happier. Whereas if they hold it under their nose, it makes them make a face that doesn't really mean anything and they don't get happier. But the truth is that this may not be, this may be some psychological research that's not replicating there very well. There's Bell's palsy. I had a set of twins in my life in high school. They were extremely difficult to tell apart. But one of them got Bell's palsy. And afterwards, her face largely came back, but she had a quirky smile, sort of a Mona Lisa quirky smile. And from that point on, nobody had trouble differentiating the two people. A disturbing photo, so we will move on here. Nuts for this figure, certainly know it. Notice, by the way, that this pathway here from the locus coeruleus with the red circle around it, what part of the brain is that going through? The one I obsess over. It's going through the periaqueduct gray. That's the cerebral aqueduct. And locus coeruleus is associated with what neurotransmitter? Norepinephrine. And it sort of has a blue color, cerulean blue, and that's why it's blue. And then you have substantia nigra down here, which is associated with dopamine. And of course, that one looks black. In this chapter, it's not the issue. This is a reward chapter, but that's associated with Parkinson's disease, loss of cells there in Parkinson's. And ventral to that is the well-named ventral tegmental area, VTA, also dopamine. And they are both parts of the ventral midbrain. And they project to a number of forebrain structures. And they aggregate, along with some of the fibers from the locus coeruleus, in what's called the medial, suggesting it's toward the middle forebrain, because we're in the forebrain bundle, which is an aggregation of sticks, and in this case, axons. And this is about self-stimulation in rodents. And this was a very hot area of neuroscience for quite a while, but it's kind of died down as the story goes. Researchers were using an open field test, so it's like a tabletop, and you can use this to assess place preference or place aversion. And they wanted to study what we covered last week, sleep and arousal and the reticular activating system. And the reticular formation, runs through the core of virtually everything you see here from the midbrain on down, even up through the thalamus. And they were aware that stimulating parts of the brain were aversive to animals. And they thought it would mess up their study if they were stimulating their brains and causing distress. So they were pre-screening the animals with electrodes in and around the reticular activating system, reticular formation. And they would stimulate the animal in a certain part of the open field and ask the question, do they avoid that part of the open field? And if the animal gets stimulated in the lower left corner and they never go in the lower left corner again, it's aversive. And the animal is excluded from the study. They misplaced an electrode and allegedly even lost the histology and never found out where it was. But they were screening an animal, and the quote was, the animal seemed to be coming back for more. So they would stimulate in one part of the open field to see if it avoided it. And not only did it avoid it, it became unnaturally attached to it. It loved it. And that led eventually to discovering reward pathways nowhere near where they were trying to study, but more in the forebrain, although I guess they could have been hitting locus coeruleus fibers. And the medial forebrain bundle is the hotspot for sticking electrodes in and getting a rat back in the day to press levers. And if you've ever seen a rat on amphetamine, they have a stereotypical look. They're hyper alert. They sniff a lot. Their head is bopping up and down. They're into it. Animals with electrodes in the medial forebrain bundle, they have the same look. It seems to be profoundly reinforcing. Now, when you talk about reward, as I said in week nothing, you always associate the VTA, ventral tegmental area, with the nucleus accumbens. And there it is, nucleus accumbens. So that, and you have had several of your peers go into doctoral programs and not follow my path and study pain, but they followed their paths and studied rewards. And they would look at nucleus accumbens, and they talk about core and shell. And I'll leave it up to you to figure out where the core is and where the shell is, and the release of neurotransmitters in accumbens. Virtually anything you know of that's fun or addicting releases relevant neurotransmitters in the nucleus accumbens. So it's an important piece of research, and it's discussed. Decorticate rage, I've done that one. Interesting, not overly relevant, I think, but PAPES, which is not Pepez, as I said for several decades. PAPES with rhymes with capes found important parts of the forebrain that seemed and are interconnected, and he thought that these were important for emotional expression. And I want you to know the PAPES circuit. And your coloring book has already preloaded some of this stuff. The mammillary bodies are visible from the ventral surface of the human brain. This is not usually where I start. I usually start with the hippocampus, and you've got two mammillary bodies, left and right, because they're not the raphe, which is midline. You have left and right hippocampi. They're very far apart, and the hippocampi send axons largely to the mammillary bodies through the fornix, and there's the fornix in that sentence. And the mammillary bodies send their axons through the mammillothalamic tract. That's mammillothalamic tract. They go from the mammillary bodies, and thus the term mammillo, to the anterior thalamus, and thus thalamic. And this is a relay. So a neuron in the hippocampus, axon goes to the mammillary bodies. They talk to cells in the mammillary bodies. Those cells send their axons to the anterior thalamus. They talk to cells in the anterior thalamus. Those cells send their axons to an area that's dear to my heart because it's bigger in liberals, cingulate cortex. The cingulate cortex is on the medial face of your hemispheres, just above your corpus callosum, and the cingulate cortex, like the hippocampus, is sort of like a big banana. It arches around, and cells in there eventually reconnect to the hippocampus. So you have a circuit: hippocampus to mammillary, mammillary to anterior thalamus to cingulate, sometimes called limbic cortex back to the hippocampus. Ba-dum-bum, you got it. Papes thought this is very important for emotion and emotional expression. We'll see, I think, in a different chapter that this circuit is very important for certain kinds of memories. And this figure here is a little bit rich in detail, but if you stay calm, have some coffee. The amygdala, by the way, is not part of the PAPE circuit, but there it is. But you'll see the paved circuit there. You'll see the hippocampus through the fornix to the mammillary bodies, hippocampus through the fornix to the mammillary bodies, to the anterior thalamus, to the cingulate or limbic cortex, and back to the hippocampus. Now, Kluver-Bucy syndrome does involve the amygdala, or what I prefer to say, amygdaloid complex. Kluver and Bucy did what, by modern standards, would be considered a fairly radical, which means kind of big, bilateral, which means on both sides, temporal lobectomy. Now, an ectomy means you remove the tissue. An otomy means you disconnect the tissue. Prefrontal lobotomies were disconnections. Bilateral temporal lobectomies means they went in, they scooped out parts of the temporal lobe. And the amygdaloid complex and some of the limbic cortex and the hippocampus, they're all in there in the medial face of the temporal lobes. And they did this in rhesus monkeys, Kluver-Bucy. They also did cats. Socially, these animals were very changed after their lesions. And we had what came to be called the Kluver-Bucy syndrome, which is over here on the right side. Bilateral, and they focus on the amygdala. Dramatic emotional changes, including reduction in fear and anxiety. Neurosurgeons are not as well regulated by federal regulations as are pharmacologists. I swear that some physicians went to Hawaii, saw a movie about Kluver-Bucy, I think cats in this case. Kluver-Bucy cats appear to be hypersexual. They mate with other cats, they mate with chickens, they'll mate with your feet. They seem to be hypersexual. Actually, they're kind of indiscriminate. And in the video, or the movie that these people saw, the monkeys that were the cats that were hypersexual from Kluver-Bucy syndrome, bilateral amygdala damage, they then did lesions of the hypothalamus. Now, the hypothalamus is not big. And, of course, the mammillary bodies are part of the hypothalamus. And they blew up part of the hypothalamus, and the hypersexual cats were no longer hypersexual. And these chuckleheads came back from Hawaii and started offering, it was voluntary to the extent that things in prisons are voluntary, they were offering to treat homosexuals for their homosexuality, to restore their natural male-female balance. We have no idea where that came from. And homosexuals were getting hypothalamic lesions to treat their homosexuality. Just unbelievably ignorant. By the way, the Kluver-Bucy monkeys were thought to be mellow and chill and not very aggressive, which is kind of true. There was, in the little land of India, if you brought your male child in and said, Billy is acting up and I have trouble controlling him, he doesn't listen, the Indian physician would say, oh, leave Billy with me for the weekend, I'll fix him right up. And they were doing bilateral amygdalectomies in children to calm them down because of this research. And that was so ignorant. The rhesus monkeys that this was originally demonstrated in, the relaxed rhesus monkeys that were non-aggressive, they were chill, they were marijuana dude monkeys from California. They were not California dude monkeys. They were socially insensitive monkeys. If there's an alpha male in your cage, and there was, and the alpha male is sleeping in the middle of the cage, you do not walk on him. but these monkeys would walk on him. If the alpha monkey were eating and the Kluver-Bucy monkey were hungry, he would just walk over and try to take the food from the alpha monkey. If the alpha monkey walked up to you and gave you a threatening stare, you're supposed to look at the floor and possibly present your rump to him. These monkeys did none of that. So what do you think happened to them all day long? Yeah, they got beat up over and over and over again. This was not therapy for anything. Okay, now we're getting to fear. And this is my thing, and there's the amygdala again. I would prefer that you start saying amygdaloid complex, by the way. And learning is very important. And by the way, let me now mark it that particularly when we get into the details of fear, one of the most important courses you can take in psychology for neuroscience, if you're going to be doing behavioral research, particularly reward or fear, is what's now called learning and behavior. Last year it was called conditioning and learning. This chapter overviews a bunch of that class. Now, when I teach that class, I don't do neuroscience, I do behavior, but it is so relevant to behavioral research in neuroscience. Let's see what we have here. So they're talking about the amygdala and fear, and yeah. Okay. Now, this figure is very important to me, and we're going to do it a few things here. So here you have a mouse in here, and you have a tone, which in the first box is not associated with anything bad happening to the mouse or rat. In the second one, the tone is associated with the grid being electrified. And having learned that, learning and behavior, this is Pavlovian fear conditioning, Pavlov. You think of Pavlov and slobbering dogs, and you don't think it's relevant, but it's the foundation of love and addiction. And in the third panel, the animal is afraid. Now, one thing I have to point out, look at that grid. It looks like a mesh, right? Where the horizontal wires crisscross with the vertical wires. Good luck electrifying that. You need two sides of a circuit. And unless you have a harness on the animal with the other side of the circuit, that animal won't be shocked in that box because that grid is not appropriate for shocking. A shock grid is typically a series of one direction stainless steel rods. And then alternate rods are alternate parts of the circuit. And the animal virtually can't help but stand on more than one rod. And therefore, when you electrify the rods, they get shocked. Now, in this box, in the second panel, we have blood pressure, which, by the way, is not a very classic way to do behavioral research, but it does exist. The next panel is much more relevant, the purple lines. Freezing. If you've ever had a rodent as a pet, mouse or rat, they are unbelievably active. They are always doing something. When I taught learning and behavior or conditioning and learning years ago, one of the textbooks I used back then bundled with the textbook something called Sniffy the Virtual Rat. And you all would get a virtual rat and you would have to run exercises. It was a great thing. Why do you think Sniffy was called Sniffy the Virtual Rat? Because rats are sniffing all the time and they're exploring and they're licking and they're scratching and they never stop. Now, mice, rats are small, and particularly when they're in open fields, and we've already mentioned an open field in terms of self-stimulation and the brain research. If an animal is in an open field, they are reasonably fearful that they're exposed and something big with feathers or big with claws is going to see them in the open field and come over and eat them. So one strategy is to get the heck out of there. The other strategy, if you can't, is to freeze. And freezing means you don't act like a rat or a mouse. You become stationary. If you've been ever trained how to locate birds or anything, it's often recommended that you pick a focal point, look at it, and don't move your eyes. And then your visual system will pick up any little movement in your field of view, and bam, you can see it. So if an animal moves, they are much more likely to be detected by predators. And if you give this animal a reason to believe they're going to be hurt, and that's why we use electric shock, because it hurts without hurting, what that means is, yes, it hurts them, but it does not damage tissue. So, you could light a match to them, but then they have blisters. And you could, hit them with a stick, and then they'd have bruises. But electric shock, appropriately done, seems cruel and unusual, but it's a momentary painful stimulus that does not have long-term consequences beyond the animal, classical conditioning, Pavlov, learning to associate this environment and or stimulus with negative outcomes. Now, your colleagues have gone to work in the labs of several researchers who do fear research. And all fear researchers do not operationally define fear in the same way. This one, ignoring the blood pressure, is freezing. And more freezing, of course, means more or less movement. Right. It's negatively correlated. You can't be moving if you're freezing, and you can't freeze and move. So that big purple bar means the animal is spending an unnatural amount of time doing little more than breathing. Even their whiskers virtually stop. One of your peers did their honors research on this. There's a researcher there was a graduate student in a lab I'll be talking about shortly. He did research showing that fear moves through the brain. It lives here, it lives there, it lives somewhere else. It has different anatomy and pharmacology depending on when you test for it. And we were doing research to try to figure out when it was best to try to extinguish the fear. And extinguishing the fear means expose them to the stimulus again without noxious stimulation and see if the fear can be reduced by extinction. Fear, by the way, is notoriously difficult to extinguish, with good reason, probably from an evolutionary perspective. How many times do you get to die? I mean, if you've survived, Bambi survives being attacked by a bear, the next bear that walks by is well fed and doesn't attack Bambi, and Bambi suddenly decides, oh, bears are chill, I don't have to worry about them anymore. No. You always look at costs and benefits. Once something almost kills you, it probably benefits you to have a healthy fear of that for the rest of your life. Every once in a while, you're going to be afraid. Every once in a while, you're going to fear. You're going to run away. Those have some relatively trivial consequences. But if that bear eats you, goodbye. You've lost the competition. Freezing and fear learning. Fear learning in general is difficult to extinguish. Freezing is a classic way of measuring fear. Your peer, who went to UCLA then for graduate school, one of the two who went there, he measured freezing in animals, both to indicate that fear had been learned and to assess whether the animals were extinguishing from fear. By the way, a second student who was only a freshman or a sophomore was sitting outside where my office was. And a friend of mine and I used to argue about statistics out there all the time. And most students looked at us like we were speaking a foreign language, but I could tell that this woman, she got it. She understood what was going on. So when my honors student needed someone to help check the reliability of his assessments of fear, she became that human. And she ended up doing her doctoral dissertation, or her master's research, her honors research on fear. And she too went to UCLA. And she used fear in her doctoral research. And she's the one who won the national award for the best doctoral dissertation in her field the year she got her PhD. And the lab she went into, and this is the name I want you to know, is the Fanzelo lab. So here's the Fanzelo lab, UCLA. I remember this is where they would have their cookouts. And Fanzelo has changed. We're all aging. There's Michael J. Fanzelo right there. Do not call him Mike. Never call him Mike. He's Michael Fanzelo. And he has an extremely productive lab. And one of his main measure for fear is freezing. One time I had to try to spend a bunch of money overnight because somebody wanted to give this Scranton $1 million. And I contacted a company saying, tell me how much this equipment will cost. And they said, I'm sorry, all of our people are tied up pricing out a grant proposal by Michael J. Fanzelo. And that was it. I couldn't get prices. And I assumed Fanzelo got his money. So that's one name, Michael J. Fanzelo, Michael Fanzelo, freezing. Now, another person who does a lot of freezing, I'll close with, but this other person is Mike, not Michael, Davis, and he was at Emory. He's now retired. And no pictures. Website. That's not the guy. Oh dear. Is he dead? So that obviously is a younger Mike Davis because this person is a colleague. Oh no, Mike, where'd you go? He spoke here, by the way. The Davis I'm looking for. All right, just trust me. This is a human being. And you'll remember, and someone sitting over to my right, if they're here, I did my doctoral dissertation on, it's Michael J. Davis, on acoustic startle. And I don't think I can scare you enough, and I won't get to see you jump. So hopefully I can inhibit barking. All right. So Michael Davis was at Yale, and he's one of the few people who not only was hired by Yale, but was also kept by Yale. He got his doctoral degree there, and he stayed. And that is not the Yale way. What Yale does is they take people in and they don't tenure them. In fact, the person I was talking about who did the research where fear moves through the brain, he was hired by Yale, and he stayed until tenure became an issue, and then he went to another lab. And Let me see. Let me try a different search approach here. Yes. Okay. So know this term: fear potentiated startle. And again, there's no picture. But as you recall, I did my doctoral dissertation on acoustic startle, and I did, lesions of the decussating fibers of the trapezoid body. But it's Halloween, and you go to a scary movie, and you know that if you're afraid, a sound that otherwise might not have affected you. By the way, some of these lesions are very close to the trapezoid body, and therefore close to my heart. And this is a picture of his paradigm. So it's very much. Oh, Kerry Ressler. Kerry Ressler was the mentor. So Michael Davis was hired away from Yale, where he had a tenured position, by Emory, because Emory had more money than God, because Coca-Cola wanted them to build an internationally renowned institute on something, and they picked neuroscience. So they had more money than God, and they went and they they hired faculty from major schools like Yale, brought them to Emory, and then those people hired mini-me's. That is, Michael Davis was mature. He hired Kerry Ressler, who was not so mature, and they started the Ressler Lab, and one of your peers became Ressler's doctoral student. And oddly, her name was Kim. First name, not last. The prior Kim was last name, not first, who went to Yale. Now here's a picture. Notice the grids here are accurate, and you condition a stimulus. It can be a light or a sound or the context, and the animal learns to be afraid. And now you elicit the acoustic startle, which we know some people in this room are able to exhibit. But if you're frightened, you have a fear-enhanced acoustic startle. And so you jump more if you are afraid. And there's the animal jumping more, and you can measure this fairly automatically with accelerometers on the floor of the cage. So when the animal jumps, the cage shakes, you quantify that. It's very easy to do. Okay, so now this is our second measure of fear. First, open field, freezing. And there I'd like you to think about fans hello, Michael, not Mike, and the researcher we're about to do after this. Then you have fear potentiated acoustic startle. It's acoustic startle. It's caused by a noise. But when you're fearful, you jump even more. When you're in a scary movie and a loud sound happens in that movie, sometimes the entire audience jumps. You can feel it in the theater. So that's fear enhanced acoustic startle. That's Mike Davis. And he started doing what I did, mapping the pathways for the acoustic startle in the brainstem. And here's, by the way, the beginnings of the amygdaloid complex. So these are subparts of the amygdaloid complex. And right off, I see paraventricular, which is related to the periaqueductal gray. Ooh, the vagus nerve. So that's the second one. And now the third human. I have to make sure my textbook hasn't decided to give up on me. Okay, and by the way, this figure down here is where we're getting to now. So the researcher discussed here, Ledoux, who is the next thing I'm going to talk about. Ledoux primarily uses freezing as his behavioral measure. Now, notice that he's talking about two subunits of the amygdala, the amygdaloid complex. But more interesting than that to me is the pathway here. So you have stimuli coming in through sensory organs, because they're plural. It can be visual, it can be olfactory, it can be auditory. And each of those would have their own pathway. Now, by the way, I just said olfactory, which I should know, because the thalamus is the great sensory relay station of the brain through which all sensory information except olfaction relays, which means one neuron goes in and another neuron goes out before it goes to the cerebral cortex. And here's the sensory cortex, which is the cerebral cortex. Notice he talks about the high road. Now, when I was a lad, there was a song that you probably never heard. Oh, please don't let this be weird. That's weird. Whoa, YouTube. I hope I don't have a lot invested in you today. So it's a famous song. And there's Loch Lomond. That's what it's about. And here are the lyrics. O ye take the high road, and I'll take the low road, and I'll be in Scotland afore you. That's the thing. You go high, I go low, I will get there first. And here you have the high road, which is relaying sensory information through the sensory cortex and the hippocampus, which is important for learning. But there's also a low road, and I'll get to Scotland for you. So the low road means that the amygdaloid complex is able to comprehend, to the extent that a subcortical area can comprehend, the amygdaloid complex can comprehend the threat of a stimulus without thinking about it, really. When you think about thinking, when you think about consciousness, you're thinking about the high road. But some things get right into your amygdala. And I do research on faces, and there's pretty good evidence that at a glance so short that if I mask what you just saw with another stimulus, you will never consciously know that you saw the face you saw because I overlay it with a different face. But your amygdala, the low road, sees that face and changes your emotional response to subsequent stimuli. This is very, and by the way, this low road, you probably have pulled your trigger by the time the high road kicks in. So information goes into the lateral nucleus primarily and then to the central nucleus. And then you have outputs, emotional behavior, which of course is what I'm interested in, autonomic responses and hormonal responses. This is Ledoux. And I've had people work with Fanslow. I've had people work with Davis's surrogate. But I've never had anyone get into the Ledoux lab, and I am hot to get someone into the Ledoux lab This guy is a really, really good and still active researcher. And he also cares a lot about communicating with the public about the research he does. And I'm about to do a lot of that. Now, notice that when you look at these three outputs, there's no anatomy there. It's just emotional behavior, autonomic responses, and hormonal responses, no behavior. Um, in the last edition of this book, I swear there was anatomy down there. And one of the three pieces of anatomy is something near and dear to my heart, the periaqueductal gray. Now, I'm going to be showing you some figures, and they may not say periaqueductal gray, they may say central gray. Well, the periaqueductal gray is central, and the term central gray refers to all gray matter at the center, and the center is periventricular. So there's central gray in the forebrain, there's central gray in the midbrain, in the hindbrain, and even, remember I asked you about lamina 10 in the spinal cord. Lamina 10 in the spinal cord is the central gray of the spinal cord because it's peri the central canal. So I did not ask that for nothing. All right, so now we're going to go to this SFN emotions lecture, and we're going to fly through a lot of these links. So here's the Ledoux lab. And there's a lot of fun resources there. He sometimes has, like one my sons did for a while, Soul Patch. He's got a song called, I think, Fearing, and I'll play that for you in a second because he also has a musical group. Let me see if I can control this. Okay, so my own brain is to me the most unaccountable of machinery, always buzzing, humming, soaring, roaring, diving, and then buried in mud. And why? What's this passion for? And he's a poet and he sings, and we'll see that. And then there's his happy lab. I don't know any of these people, and I probably never will because you guys won't go work with him. The higher order theory of emotional consciousness, visual cortex, subcortical survival circuits, which undoubtedly include the amygdaloid complex, long-term memory. Here we go. So here's freezing. Oh, and I didn't mention this one. Active avoidance. He is trying to bring avoidance back into neuroscience. When I was avoidance was a very big field. And in learning and behavior, we spent a lot of time on avoidance. But, there's avoidance. You have a two-chambered box with an obvious divider. And on one side, bad stuff happens. And on the other side, bad stuff doesn't happen. And if you're a well-trained animal, you avoid the bad and maximize the good. And there's the freezing, and there's some neuroanatomy and condition stimuli, classical conditioning. You all took fundamentals of psychology, I think, CSUS, Bell, meat powder, and there's the amygdaloid complex. So the birth, death, and resurrection of avoidance, a reconceptualization of a troubled paradigm. Oh my God, I was so excited to see this. I actually haven't read this article yet, but I was in the field when avoidance was dying. But it's back, baby. So using neuroscience to help understand fear and anxiety, a two-step framework. A lot of good stuff here. And this is one of his books for the popular press, 2015, Anxious by Joseph Ledoux. It's a good book. It's based on solid science. And by the way, I started to mention and then got distracted. I saw him give a talk at Neuroscience about the amygdala and the high road and the low road and the anatomy of the amygdala. And when I came out of there, I virtually stop saying amygdala. It's the amygdaloid complex. There's so much going on there. It's not a thing. Um, there's okay. If you're into molecular neurobiology as opposed to behavioral, fear memories require protein synthesis in the amygdala for reconsolidation after retrieval. This blew me away when I first discovered this reality. Usually, when people are doing therapy for people with fear, they try very hard to not reactivate the fear for fear that reactivated fear will lead to more learning and more fear. But there's something weird here that when a fear memory and possibly other memories are reactivated, they have to be relearned. And that's the term reconsolidation. They are brought out of storage, they make you fearful, and then you have to put them away. And they are vulnerable to disruption while they're out of the box. And a lot of research, including Mike Davis, he thought he had a drug that was used to treat hypertension that, when combined with behavioral therapy, was really powerful in terms of treating PTSD. 1 year at Neuroscience, he had a full-fledged film crew with him, multiple cameras, gaffers, the whole thing, because they were going to go public with this therapy procedure. I don't know whatever happened to that, but reconsolidation, know the term. Consolidation is what you do with memories. You put them in short-term memory, you move them to long-term memory. That process is called consolidation. Well, now we have reconsolidation. So activating a fear and doing something to disrupt either the experience of that fear by giving you a drug that blocks your autonomic nervous system, or possibly disrupting protein synthesis. We have new ways of dealing with treatments of fear. And there's reconsolidation again, and there's extinction. Again, classic learning theory. Lateral amygdaloid complex, fancy stains, scanning confocal, no doubt, and fluorescence. Oh my God, so beautiful. Look at the figure on the left. Now, unfortunately, he labels it. What stain is that? That's Nissl, my friend, and boy, it's a beauty. It is a beauty. And notice that the amygdaloid complex is a complex. Now, next to that is a stain that those of you in the lab will see in one of the atlases we use. That's an acetylcholinesterase stain. And acetylcholinesterase is what breaks down acetylcholine. And if I told the story about making popcorn with an acetylcholinesterase inhibitor, that inhibitor was in the lab for that stain. And you can see that not all subnuclei of the amygdaloid complex are equally blessed with acetylcholinesterase. Not even close. And then there's a third one, which is, so the first one, Nissl, is a cell body stain. Second one is cell body, but primarily if the cell bodies contain acetylcholinesterase. The third one is a fiber stain, silver. And there's the fear again. So there's classical conditioning, and there's the amygdala on the amygdaloid complex on the left. And notice that once again they get the grids correct. And notice the histograms: CS on, zap, and then CS on, no zap, but we're testing you. Okay, so obviously I'm a big fan of Ledoux, mostly because I've never gotten anybody to work with him. So this is a link to the birth, death, and resurrection of avoidance. We don't need to see that now. I hope YouTube is working now. Okay, this is his musical group. If you live in the city, they play in the city. And apparently they play every year at Neuroscience at a musical event that I've missed every year because I go to a different. He looks a little creepy. And I bought their album, and I will play a song from the Amygdaloids in a second if YouTube is working. Oops. Oh no, where's my link? All right, let's go back to this one. So the song is all right. We'll just do it the old-fashioned way. And if you didn't think he was creepy before, and this, by the way, looks like Pennsylvania to me.
While I was fearing it came, but with less of the fear, because fearing it so long had almost made it dear. There is a fitting dismay, an appropriate despair. 'Tis harder knowing that fear is due than knowing it is here. The waiting is the worst. It ties you up in knots. Anticipation a curse. 1,000 empty shots.
Yes, I own that album. Okay. Here is a talk that we will not watch, but it's a discussion, Big Think, which means it's not very long, and it's by Ledoux, Big Think. More on emotional consciousness, more on the emotional brain. Now, oh my God, look at the figure that differentiates between dorsal and ventral PAG. Now, remember, I gave you a lecture on dorsal and ventral PAG, largely, and it dealt with non-opioid and opioid stimulation-produced analgesia. Where's the figure? Okay, so you have unconditioned threat, conditioned threat. Unconditioned means basically free with the genes. You don't have to learn it. It just is. This is learned. Look at this, baby. PAGD for dorsal PAG. Oh my God. PAGV for ventral PAG. Bam. And notice that they end in defensive behaviors. Somebody go work with this guy. So here's a discussion of, or not, just a link to reconsolidation. There's fear, and there's the high road and the low road. But why we're no, it's not here yet. Notice here's a behavior that is he's very fond of. Blood pressure I don't care about. Hormones I don't care about. CG, that's central gray, and I've already lectured about this. It's in the midbrain. The central gray is the PAG. That's freezing. And lateral LH is lateral hypothalamus, and that's blood pressure. And paraventricular nucleus, paraventricular nucleus, it must be a nucleus near a ventricle, and hormones. And more on learning. I don't know if there's anything I'm really after you learning here. So again, CG here is generic. Central gray of the midbrain is my beloved PAG. So that's in here somewhere. Freezing, yeah. Oh no, here it says paraventricular gray. So it's all spelled out. Okay, where are we on time? Not a lot of time. And if you've had any trouble loading this up, I think a wise person would stop now. So I'm going to try to stop. I'm about to lecture on the stress version of dorsal ventral opioid, non-opioid, which I will do on Thursday after the quizzes. So on Thursday, read ahead for the chapter we've been dealing with, quiz on last week, and then I'll try to wrap it up. Hope you're having a good week. Bye.